So here we're going to look at the light reactions of photosynthesis. So here in the chloroplast, which is a double membrane organelle, we have something called a thylakoid. Now this is the location of where the light reactions are going to take place. Now in the space around the thylakoids, a stack of thylakoids is called a granum. And then the space around the thylakoids is called the stroma. The stroma is where the Calvin cycle happens. So here, this two green lines right here represent the double membrane of the chloroplast. So we're going to zoom into the thylakoid. Now to show you a visual of the relationship between the light reaction and the dark reaction, here you can see the light shines down onto the thylakoid where there's these things called photosystems, which are the purple boxes in this picture, and then you have the electron transport chain. So basically the light reaction is going to happen in the thylakoid, and the products of the light reaction are going to be a reduced electron carrier, NADPH, ATP, and oxygen as a waste. Then the Calvin cycle occurs in the stroma and will fix carbon dioxide from the atmosphere, from the air. It will invest the electrons and hydrogens from the light reaction as well as use the ATP in a series of endergonic reactions in order to build G3P, which is going to be the building block for life. So in the light reaction of photosynthesis, there's this molecule called NADP+, which is the final electron acceptor of the light reaction. So this NADP+ is an electron carrier that at the end of the light reaction, it gains or pulls the electrons down and also gains a hydrogen. So right here, this is a reduced electron carrier, and it will literally carry the electrons over to the stroma to be used in the Calvin cycle, where it will be oxidized. Okay, so let's go ahead and look at this light reaction. So within the thylakoid membrane, this line right here represents the lipid bilayer of the thylakoid. These purple rectangles represent photosystems 2 and 1, which are basically proteins, and that's where you're going to find your pigments. So the main pigments in plants are chlorophyll A and chlorophyll B. And in the middle of chlorophyll is the porphyrin ring with this magnesium right here in the middle. So in a little bit here, we're going to see how electrons are donated from the chlorophyll molecule. They're actually breaking off of this magnesium. Okay, so here in between the two photosystems, we have an electron transport chain made of enzymes. So here's our PS2 and 1. PS1 was discovered first, and PS2 was discovered second. So when we look at how this works, light shines down, and there's pigments housed within the photosystems. They're going to absorb and reflect light. So the light energy that's absorbed into the photosystems will excite the electrons to a higher energy state, breaking them off of chlorophyll, and now they can enter into the ETC. So this happens for 2 electrons. And so now, at the end of the electron transport chain, here is that electron carrier. So as these electrons flow, this is called linear flow, where you would find the electrons flowing from photosystem 2 to 1, and then ultimately to the electron carrier. Now sometimes, over here, let's see this play out. So then these ones will flow, reducing the electron carrier. Now as the electrons flow from PS2 to PS1, they pass electrons down, the enzymes do, and they pump protons into the thylakoid space. This green area is within the thylakoid. So now those electrons that were donated from chlorophyll B right here that ended up on the electron carrier are being replaced by these electrons flowing down the electron transport chain. But where do we get the electrons to replace in chlorophyll A? That's going to be from the splitting of water. This process is called photolysis. So you can see right here how the water molecule splits. So now these electrons will replace the ones that were just donated into the electron transport chain. Oops, that was on a timer. And then the oxygen will actually pair up with another oxygen, forming O2, which makes it a non-polar molecule, so it can actually diffuse out of that, through that lipid bilayer of the thylakoid membrane. Now this leaves the two hydrogens from water still here, and because hydrogens have, or protons have that positive charge, they cannot leave by diffusion through that thylakoid lipid bilayer. So here we go. This continues to happen every time the light shines down. All right, so over time, you get an accumulation of protons within the thylakoid space. Here we're establishing a proton gradient, and we're making what's called a low or acidic environment, a low pH, within the thylakoid space. And it's this high concentration of protons that leads to a process called Kamiyas Moses, where the protons will flow. And basically you can think of it as like facilitated diffusion. They're flowing through a proton channel, and a protein channel, and as they flow through down their electrochemical gradient, ATP synthase is able to join together ADP and an inorganic phosphate to make ATP. So this diffusion of ions is called Kamiyas Moses, and it's how we're able to produce ATP. So at the end of the light reaction, we have, this is photophosphorylation, this process here. So at the end of the light reaction, you have your ATP and your reduced electron carriers that will both go and be used in the anabolic pathway of the Calvin cycle. Now I really like this picture simply because it shows the lipid bilayer, which explains why protons are unable to diffuse through. That charge on them will keep them trapped in the thylakoid space until they can flow through ATP synthase. This lipid bilayer is also why O2, a nonpolar molecule, can just diffuse out. And that is the light reaction of photosynthesis.